presented by Dr. Ji-Yong Park, radiopharmaceutical chemistry development. Please welcome Dr. Park.
Hello, my name is Ji-Yong Park in chemistry team, our department. So I will talk about this radiopharmaceuticals and development based on the chemistry base. So today, after we talk and discuss about some possible structure and possible agent about the theranostic aspect, especially on our nuclear medicine field. So we already discussed about some cutting-edge radiopharmaceutical for the therapeutic uses one from Dr. Sa's presentation. So I just prepare some basic one and how we can understanding the concept of the Theranostics, and then what kind of radioisotope is possible to use to diagnostics, and then we discuss about some chemical structure for the use this type of concept to our radiopharmaceuticals. The radiodiagnostics and radiodiagnostics is some combination words from the therapy and diagnosis. So originally the diagnostics or diagnostics concept is already studied from the others several fields of science. For example, some basic science and nanoscience and optical imaging and laser ablation and surgery, we so-called opto-diagnostics already studied. And in our field, we use this kind of concept to our study. So you already know that for examples. For example, we use some radioisotope based one. It means that we just injection about using iodine or technetium for the diagnosis or therapy, and using some fluorescence based isotope labeled chemical was used for the bioimaging guided radiotherapy. Traditionally, we use like this concept of radiopharmaceuticals. It means that already exist radiopharmaceuticals are conjugated to the radionuclide, and then we use these kinds of drug for the diagnosis or therapy. A radioactive compound uses for the diagnosis and therapeutic treatment of human disease. So because of it was really well defined and really small amount, it means that the trace amount of the radionuclide, so it is safe and non-toxic for human administration. Some really impressive things of nuclear medicine is many of the agent was used. Many of the agent is use the metabolic pathway for the imaging. For example, FDG and iodine and phosphonate is capitalized example of this. So using like this concept, it means that we use the metabolic pathway. So some kinds of radioisotope added one is possible. Diagnostics are radiopharmaceuticals. There are some kinds of radiopharmaceuticals already exist for the radiotherapy. So we use only radioisotope is one case, and radioisotope and ligand chelated one is an example, and drug conjugated to the radioisotope is one example, and ligand and linker and radioisotope is some example. We already discussed about some kinds of radiopharmaceutical type of this from Dr. Su's lecture, the prostate cancers one or Thera is some example of this. Nowadays, some really new kinds of radiopharmaceutical is emerging in this field. Some nanoparticle is possible to use some diagnostics because of nanoparticle have some natural property. For example, some specific modality. We use the kinds of property to some specific target region, and adding some radioisotope, we can finally have some property of diagnosis or therapy. Moreover, nowadays some drug was already loading to the kinds of the radiopharmaceuticals. This is some example of nuclear medicinal radiopharmaceutical. We already know that the glucose is a main energy source for the cell, so we easily exchange the hydroxy group to the F18, and then using some metabolic pathway, we can easily imaging of the tumor cells. This is some mechanism of how glucose was uptaken to the target site and accumulated. The original glucose was uptaken using glucose transporter, so-called clot, and then glucose goes through the enzymatic pathway, and final product is carbon dioxide. It was directly diffused to outside of the cell. But the F18 exchange one is also using some same transport system clot, but the enzymatic process is not occur because of some, this site is not this site is not possible to make some enzymatic pathway, so it was easily accumulated to the tumor, and we can imaging easily. This is some example of human. Left side is the normal image, and right side is a patient. We can easily see the spots some pathological region. Because of we understanding some cutting edge radiopharmaceutical, especially some field of diagnostics, we must understanding some traditional method of diagnosis and therapy. A really well established example is technetium labeled one. Technetium have some half-life 6 hours, and it's emitting only gamma ray. So because of it was easily generated from the generator system, so we can access easily and labeled to these kinds of radioisotope to some specific target ligand. More interesting thing is the rhenium labeled system. Because of rhenium is relatively similar physical property compared to technetium, so we use rhenium as a therapeutic radioisotope simultaneously injection to the human, because of it was using same kinds of character of technetium. Its half-life is 16.9 hours, and it's emitting gamma ray and also emits some beta particle. So we use the beta particle for the therapeutic efficacy, and its production is really good for using because it is also generated from generators. It is also made from the to the system. This kind of radiopharmaceutical must prepare like this form in this key formation. In that, some ligand is already contained, and we adding some reducing agent and stabilizer as already exists in the vial. So we use some generator system for production of technetium. So we can easily adding some this kind of radiopharmaceutical to the key formation and just to making some labeled one using stirring, and if needed we can add this some stabilizer, and after labeling step we must check the TLC for the QC controlling. This is our facility for the labeling of technetium, and lab have some clean hood, and then the generator system is here, and we can check the TLC and QC in like this well shielding hood, and shaker is for the labeling, and the heating bath is also some kind of labeling condition for radium, like this radioisotope. So we discuss about some physical property of the technetium and radium, and then we check its imaging and its chemical property. So this is technetium chemical structure that is really similar with the radius, and it's charged to iodine, so it is well uptake to the thyroid and gland. It was relatively same, the pathway of the iodine. So normally technetium is used for the bone imaging because of some kinds of agent was already existing, like this phosphonated form. This phosphonated form is some kinds of ingredient of making some bone. So we use like this mechanism. The technetium is easily labeled in this site, so we can image it of the bone. So after labeling, we can injection to the human, so we can easily make some like this bone image. And DTPA is a example for the kidney imaging, because you can see the different kinds of functional group of the end site of the chemical. So its functional group is really hydrophilic, so it was go directly to the kidney right after injection. This is an example of the kidney's creation using technetium DTPA. And I already told you about the same physical property of the radium and technetium, so we can easily access to the same chelator for the labeling and diagnosis or therapy. Radium DTPA is also can be prepared the same pathway of technetium DTPA, but the labeling condition is quite different. For the labeling of radium, we must use some boiling system. Or the system is relatively same, but the final labeling condition is using some water bath for our boiling, and we can take some 2 hours for our labeling condition, and we can use this kind of hot form for the labeling for the imaging or therapy. So I already told you the same kinds of chelator or ligand is possible to use technetium 99M and radium. Using some technetium labeling system was widely used as imaging agent for bone disease, and using the radium HEDP, we can use it as an effective palliation of bone pain. The other example of using technetium and radium is like this colloidal form of radiopharmaceutical. macroaggregated albumin and sulfur colloid and tin colloid, antimony sulfur colloid is an example. Because of each labeling step of labeling method of intrinsic labeling of the technetium and rhenium, it was really stable in our body. So its main target is lung or liver or spleen and lymph node because of its size. So right after injection, it was go directly through the body's organ, and then it was accumulated easily, and it is really not easy to excretion because of its size. So we can images of lung and liver, spleen, or like this lymph node. This is example of technetium sulfur colloid, and you can see clear image of the liver. And using antimony sulfur colloid, this star shape is an injection site. We can easily take some image of the lymph node. And this is really interesting image of the technetium and rhenium. We use same structure and different radioisotope for the different application, but the image is relatively same, but its application is one thing is diagnosis, the other things is therapy. Because of rhenium is emitting gamma ray and beta particle also, so its imaging quality is not good compared to the technetium. This is a human study of the technetium and rhenium, and relatively same image, but the image quality is not good using for rhenium because of the beta and gamma is both emitting from the rhenium. So some interesting example of using rhenium is the repeater study. Because of its structure property, you can see the long alkyl chain. It means that its property is really hydrophobic, so it is well addicted to the liver. Using like this physical property, we using some Chelator and N2S2 for the labeling of rhenium, and then after labeling, we injected it to the mouse using IV. So you can see clearly to uptake to the liver. And then we use like this radiopharmaceutical for the multi-center trial for liver cancer. This is one example of the radiopharmaceutical using drug. Using paclitaxel labeled, FAT labeled paclitaxel, right after injection, we can see the targeting site in here. This is one example of the diagnostics. The last things we talk about some diagnostics for labeling some metal radioisotope is the gallium. Because of gallium is some capitalized example of diagnostic radioisotope, because it's really easy to access because of generation system, and its half-life is 68 minutes, and the one generation system is possible to use over 6 months. So we must know some chelation system of gallium. Normally we use some chelator for the gallium is Ga-NOTA. Right after labeling of Ga-NOTA, we want to find some we wanted to find some competition of the other kinds of metals. So after labeling of gallium, we added some different severe concentration of the iron solution and copper solution and indium solution and gallium solution and magnesium solution. So after adding some 0.760 millimolar of copper, the about 13% of the gallium is not labeling to the Ga-NOTA. It means that the copper is good for Ga-NOTA labeling. It was competition using of gallium. Indium is also have some really good for labeled with NOTA, and iron is also. So in like this kinds of method, we must find some adequate peptide and protein or nanoparticle for using some like this bifunctional chelating agent. So we wanted to use some small molecule. We must know some coordination chemistry because of some bifunctional chelating agent is already used for the labeling. So we already know about some like this technique is indirect and through indirect method for the labeling. So if we want some labeling of peptide or other small molecules and other ligands, and using some covalent linkage and the bifunctional chelating agent is here, and so have some other labeling condition, we can easily labeling some like this kinds of radiopharmaceutical. So important thing is we must know about the stable complex of these kinds of metal radioisotope to the bifunctional chelating agent, and then we must know about proper labeling condition. For example, pH and temperature and reaction time. So there are many kinds of bifunctional chelating agents already exist. So we must know the stereochemistry of this kind of chemical for the labeling some different kinds of radiometal. So we already discussed about some kind of example of peptide insert from the Dr. So's lecture. So, and the other example is using some small molecules for the labeled of gallium, and peptide and antibody is also studied to use like this bifunctional chelating agent system. Nowadays, we use some nanoparticle for the multimodal imaging, and the use 6-8 gallium is also used for the diagnostic imaging. This is one example of the gallium NOTA RGD for the diagnosis. So the RGD is well interaction with some angiogenesis factor, so we can easily make some image of angiogenesis imaging using like this chemical. More interesting things is that we use some linker as a isothiol linkage because it is really stable to our body. And then the chelator system is we use NOTA for the labeling of gallium. So the targeting image is here, red arrow. Without cold RBD, we can check the clear image of a tumor uptake to here. But with cold RBD, it means that the blocking study, we can see the faint image of the tumor site. Last thing is consideration for radionuclide therapy. Because of radionuclide therapy is using some radionuclides, so we must understanding some kinds of diagnostic radionuclide or therapy radionuclide. Because of it is totally different from external radiation therapy, we must know the mechanism and pathway of internal therapy. And some kinds of metal radioisotope was used with or without carrier, so we must know its exact biodistribution. Before we use some radioisotope to the radionuclide therapy, we must understanding well about radionuclide, and then we preparing some adequate targeting, and we check the effectiveness of the targeting system, and we choose some specific carrier. For example, some chelator and bioactive molecule is a must selection for the specific pathology, and then we understanding some dosimetry because we use some special radioisotope for the therapy, and cost is one kind of consideration. This is an example of already uses radionuclide, and you can see the iodine. It emitted some beta particle and gamma ray So it is possible to make some image of using spectroscopy and possible to therapy radar stuff using beta particle. And yttrium is only emitted some beta particle, so it is only used for therapy. And lutetium is emitting some beta and gamma, so it is also possible to use diagnostics. And rhenium is also possible to diagnostics. This is the last slide of my presentation. So I think that the most important thing is we must know about some specific chelator for using some radionuclide. Using EDTA system, we can easily labeling our technetium for the diagnosis, and we can access to the rhenium labeling for the therapy. And the TPA is relatively same reaction condition and relatively same possibility to labeling of the radioisotope. More interesting things is the NOTA and DOTA case. We already discussed about the labeling condition of DOTA. Because of the stereochemistry, the DOTA needed some really harsh condition for the labeling. Normally, we use the boiling system. But in the case of NOTA, some labeling condition is really mild. It means that the room temperature. So we can easily access to the labeling using gallium and copper and lutetium. So there's some kinds of study is already done using DOTA system, but we expect that some kinds of radiopharmaceutical would be exchanged to the NOTA system. So the meaning of the chelator is that we must be understanding some chelation system, and we must know some specific condition for the labeling, and we choose appropriate radioisotope for each kinds of application. This is the end of my presentation. So if anybody have some question, freely writing some chat box. Thank you.
So this is Lee again, and before going on, waiting for the questions. As Dr. Park explained, the previous achievement by Jae-Min Jung, the rhenium-188, was actually picked up by Jae-Min Jung 2 or more decades ago. And by just recognizing the similarity between the technetium and rhenium, however, the slight differences were just kind of interesting. Would have been interesting to Dr. Jae-Min Jung, Professor Jae-Min Jung. So we were baptized very early by the possibility of rhenium compound, but rhenium is a generator driven one, and Rosnett from Oak Ridge, he let us have the tungsten rhenium generator for such a long time, and later on we purchased, and however we found that the continuous demand from the clinical people, if that kind of demand is not guaranteed, we cannot continue to keep the rhenium generator in our the clinical situations. And then the rhenium, the attractiveness of the rhenium had been decayed. So it was a shame. The IAEA was interested in propagating the knowledge of, about the use of the rhenium in addition to technetium. And technetium and rhenium combination was the kind of for some time it seemed to be a prodigy. and hope for the future, but it was not. And Dr. Park, and let me just kind of convey the interesting and stimulating news. Yesterday we did the same workshop with Bangladesh, and one of the best radiochemists from Bangladesh is now joining, and he would have enjoyed the different advanced version of presentation by Dr. Shaw. He is a good radiochemist, so he really wants to know the progress of the Malaysian colleagues. And then I am going to, I have invited the Myanmar colleague who had been a international fellow here several years ago. Dr. Su is from has logged on and from Yangon. And as is already told, there's a so there's a question. So Dr. Park, I just kind of fill the gap. So would you please just while reading the question, or Dr. Fakrazi, so would you please explain your question as a directly?
Yes, I see the question.
I can answer that thing because of Dr. Park's explanation is somewhat confusing because of that is the after making gallium complex. He explained that after making gallium complex, he added some solution, but actually we did a competition of the gallium labeling. So in the solution that contains metal ion, and we added gallium-68 into the chelating system. So that is a competition reaction with the gallium-68 and other metals. So after making Ga-NOTA complex, even though in a 6 molar HCl solution, even after the 4 hours boiling, there is no decomplexation. So it's extremely stable. So if making gallium complex with NOTA, there is no chance to demethylation in vivo system. Okay. So just a competition study, not after complex formation. We had a solution. It is not this, that procedure. So it's just mixed with the gallium-68 and other metals. Okay.
이윤상 교수 말씀하시는 게 굉장히 마이크에서 멀게 들립니다.
아, 그래요? That experiment doing with after making gallium complexation, he added a solution, but it's not true. Just we mixed with the NOTA and gallium-68 and other metals. So we checked the gallium labeling efficiency with the competition assay with the other metals. So as I mentioned that after making gallium NOTA complexation, even in a high acidic and boiling temperature, there is no evidence with the deep complexation from that complex. So it's highly stable. So even in vivo system, we can expect that there is no demethylation or decomplexation reaction in vivo. Can you hear me? Okay. Thank you very much.
This time it was clear.